So the authors of this paper detail a new Rosetta application for the prediction of protein DNA, protein RNA, and RNA complexes. They mention that although deep learning has been very successfully applied to protein and protein complex structure prediction, protein nucleic acid complex predictions are still quite poor as of 2023. I should note that this may not be the case anymore with the release of AlphaFold 3, which can also predict protein nucleic acid complexes. This paper was released in 2023, so I don't know how RoseTTAFoldNA compares to AlphaFold 3. So this RoseTTAFoldNA is actually only a minor upgrade to RoseTTAFold. It was trained on the same PDB data that RoseTTAFold was trained on, except it was augmented with all RNA, protein RNA, and protein DNA complexes in the PDB. So the model architecture is also very similar. RoseTTAFold has a 3-track architecture with each track refining 3 macromolecular aspects: sequence, 1-dimensional, residue pair distances, the 2-dimensional aspect, and atomic coordinates, 3D. RoseTTAFoldNA's architecture simply added elements corresponding to nucleic acids to these tracks. For instance, tokens for the 4 DNA, RNA nucleotides were added to the 1D track, and the 3D track added just 3D representations of nucleic acids. So in total, RoseTTAFoldNA has 36 of these 3-track layers with 4 additional structural refinement layers. So RoseTTAFoldNA was validated on 224 monomeric protein nucleic acid complexes, and it performed very well, as indicated by an average LDDT, local distance difference test, score of 0.73. This is a similar metric to PLDDT and to RMSD in that it measures atomic deviation of a model. However, the scores range from 0 to 1, and 1 is the perfect score in this case. 45% of the models produced by RoseTTAFoldNA had greater than half of their native contacts, so those were very well preserved. And the authors mentioned, however, that RoseTTAFoldNA doesn't do that well for complexes involving large nucleic acids with more than 100 nucleotides. Notably, RoseTTAFoldNA also showed a very decent ability to distinguish binding and non-binding DNA sequences for transcription factors. And it also performed well when predicting the structures of RNA complexes alone without any protein. There it also achieved a mean LDDT of 0.73. And it did well even when there weren't any homologs or there were very small numbers of sequence relatives. So compared to other tools, how does it fare? In terms of pure protein structure prediction, it achieves very comparable TM scores to AlphaFold 2, being built off of the same architecture, essentially. In terms of RNA structure prediction alone, it performs ever so slightly worse than the top-ranked deep learning model for RNA structure prediction. However, in terms of protein nucleic acid complex prediction, it's something completely novel and in uncharted territory since no deep learning model exists for this purpose as of 2023. However, AlphaFold 3 has been released and that will need to be compared to this if it hasn't already.